Hello everyone. In this video I will show you how to import and analyze a set of patients that were sequenced on a Roche 454 machine for CFTR. So the first thing is we will import the CFTR gene as a reference to align against. You just need to type CFTR and it will download the gene from Ensemble. We can also define a region of interest on which we will be able to search for variants. So as it's only one gene, we will also use CFTR, but we will just look at the translated exons and include flanking sequences in the introns of 20 nucleotides. So now we have the references and regions of interest. We need now a few patients. So we'll create just 4 patients. There are more in the data set, but this will be able to show how it works. Next we'll need to import the data files. So it's a 454. So there are 2 files. One is a raster file. The second one is a quality file. We will do some basic quality checking to have an idea of how the run went. So here we have the graphs for the length distribution. So most of the sequence reads are below 400, 450 as a quality distribution. So most of the reads have a very high quality. The beginning of the sequences are overrepresented. That's normal. That's part of the barcodes. The quality of the reads on each base. So anything beyond 450, 500 has a lower quality. This is the base distribution, and we also have the GC content. If you'd like, you can of course also print out the report. Just click on the report button. Then we need to import the barcodes. We want to make sure that all the barcodes are correctly identified, so we'll prefer to reject any read errors in that area. As we've seen that anything around 450 and beyond is not of high quality, we will reject those reads, and we'd like at least a minimum of 20 average quality. We will trim on the 5' end by around 20 nucleotides, and 3' is a bit longer, so we'd rather trim 35, and anything beyond 450 will be cut off. So now we can associate the reads with every patient. So the first tag goes to patient 1, tag 2, patient 2, etc. We'll ignore the other patients. So now we have one set of read for each of the 4 patients. Here on the left hand side you have the patients, the reads. So now we want to align against CFTR. You can choose different aligners, but we'll take ours for the moment. So we'll take the default parameters, and there will be another video to show how the parameters actually work. You need to do that for all of the data sets. So take the next one. It keeps the parameters from the previous run, so you don't need to worry about that. Just click run line. Same for the last tag. So now you see we have the tags, we have an alignment set and the genes that were found, so we just can now search for variants in all of those genes. Click the scan button, So we'll have also several parameters, but we'll keep the frequency at 25% minimum, coverage at least 20 reads, and we'd like to see only variants in the regions of interest, so we check that box. We now have several variants on the gene. We have a few filters that were already set in a previous run, so we still would like to see only heterozygous, so the frequency should be bigger than 35% and smaller than 65, and we'd like to see only things that have a minor allele frequency below 1%. We can also check whether variants have been seen before online on the Ensemble and DB-SNP database and 1000 genomes. We can sort by position, so we see that certain pseudo variants have been seen several times on each patient. That means they are most likely artifacts. Those are deletions. That is common on Roche sequencing equipment. Then there is one variant that has been seen before, that is annotated important and probably damaging. So that is one that we would like to see. We right click. We can visualize now the data. Here we have the reads, and we can see that the variant has been seen in forward and reverse roughly equally. Also, it's a very nicely heterozygous position, so that is most likely a real variant. If you right click on it, you will get the positions on the different transcripts. You can also annotate it as a mutation, for example, and you can then report the variant. You can type any name. There's a patient ID which goes with it, and you can prepare a report which you can basically save locally in the database. You can also export as HTML. Which looks like this. So you have the variant with the HGVS nomenclature on the C dot, P dot, the transcript ID, the frequency that it was seen on. It has been stored twice in the local database. It also has a RSID, and of course you get the information of the coverage, which is important to make sure that all the exons have been correctly covered. And if you go back now to the main screen, you will see that you have now a patient, which is probably patient 3, where you have attached the information that this is a mutation. So you can actually build up with various tests the variants on each patient and build a patient-centric database of the variants. This concludes the first video. Thank you very much.